And now, without further ado, I would like to pass the word to Judith Campisi, who requires no introduction. I think she is one of the most well-known people in the industry. She came up with the first senescent cell hypothesis. She's a professor at the Buck Institute and a member of the National Academy of Sciences, I think National Academy of Medicine as well, and is one of the most prolific people in the field. There were companies started based on her research, and I guess the entire field of senolytics was invented and ignited by Judith. So Judith is going to talk about senolytic drugs, finding the right targets. That's the most important part. Judith, the floor is yours.
Thank you. Thank you, Alex, for this kind invitation. Let me try to share my screen. Let's see. Okay. Can you see my screen? Is this?
Yes.
Okay.
Now let's go into presentation mode. Does it help?
I am in presentation mode, I thought.
Nope, just you already shared the screen, but click the presentation mode on your PowerPoint so we can see the full slide.
Yeah, I did. Let me go back. Hold on. Maybe I have to do it.
Yep, perfect. Perfect. It was almost perfect for a second, but now it came back.
Okay.
So let's do it again. Just click presentation mode, or if you go to view and full screen, that might be also a possibility, or slideshow and start slideshow.
Oh dear. How's this? No, that's not working.
Well, slightly better, but still, there is a small button down there right next to, yep, almost here. To the right, no. This one, yep.
Yeah, that's what I did last time.
It doesn't work for some strange reason. Yeah, so
all right.
We've seen this before.
Yeah.
It's possible to fix this. And of course, with your presentation, we don't want to have half measures, right? So two ways of doing it. We can convert it to PDF and show the PDF document. So one of the ways is to basically save it and do it like this, or present it by speaker full screen. Let's try it doing this.
Yeah,
this
let's try this. See if that works. No.
Didn't work. Can we just very briefly try to convert to PDF and see if it can work? Or Morten, maybe you have other tips and tricks.
Judith? Did this work?
Judith,
yes.
And Judith, just go to slide 1. We can see the slides and read what it says. So if you can go through them like you have them here, maybe zoom in a little bit on the slides, that's fine. Then we can.
Yeah, but Morten, let's try and do this one so that it could be, recorded and shared. So it's all half measures for Judith's talk.
Well, I can send you this afterwards if that will help.
Okay, it depends on how you want to do it. So two ways. One is we go to save as PowerPoint, open at this PowerPoint.
Judith, we have it here.
Perfect. So it was just waiting. So now let's just reverse the screen. So there is just switch screens.
Swap
so
displays.
We can see your swap displays.
Up in the top, if you click up in the top, there's a button that says swap displays. Up in the top of the screen to the left.
Okay, but I have to...
Can you see the swap displays button? Just click it.
Yes. How is that?
Perfect.
Great. Judy, that is a miracle. Thank you so much for staying with us. No half measures. You are perfect. We can see you very well. We can see the slide. Let's go. You are the last speaker of the day today. And yeah, the highlight.
Thank you so much for this invitation. It's really a fantastic meeting. This is the disclaimer. that I'm a founder of one of these companies. There are now probably 100 companies that are doing this. But let me start with what we all know about aging, which is, of course, nobody dies of good health. We die of diseases. And the diseases are very different from each other. However, what tends to happen is that they all have the same trajectory over the lifespan, meaning very rare in young people, and they go up approximately exponentially with age. So of course, those of us who work on aging think this is not a coincidence. We think there are basic processes that are driving all of these diseases. And of course, cellular senescence has now become a pretty good candidate for a basic aging process, certainly in mice. We still don't know in people. So before I talk about drug discovery, which I know is the focus of this meeting, let me remind you about some important aspects of cellular senescence. So the first is that the phenotype of senescence is tripartite, meaning there are 3 modules. They often go together, but they can be dissociated from each other. And when all 3 are present, we feel confident in saying this is a senescent cell, but there's now a lot of queries as to whether, for example, if a neuron develops this multifaceted secretory phenotype, do we call it senescent, given the fact that it's already irreversibly growth arrested? So these are questions that the field is now grappling with. I don't have clear answers, but stay tuned because I think it will shape how drugs are designed to try to intervene in the process. So the first thing which you all know is that there are bad things that senescent cells do because they are caused by many cancer-causing and actually age-causing stimuli. And this includes things that damage the genome or the epigenome. Even though this growth arrest is a potent anti-cancer mechanism, many oncogenic mutations then induce this phenotype. But things that derange metabolism, for example, mitochondrial dysfunction, things like high glucose, the development of these ages, they all can drive cells into this senescent phenotype. So we think all of these are bad, all of these happen with age, and they all develop eventually into an aging organism. And so we would like to prevent this phenotype for that purpose. However, it's really important to remember that the other half of the senescence response is good for you. And so I'd already told you that the growth arrest will inhibit tumorigenesis. Many of these secretory products will also fine-tune development during embryogenic development. Many of these secretory phenotypes also fine-tune parturition. So in mice, the secretory phenotype dictates when labor will occur. In humans, there's no cause and effect experiments, but for sure, women who undergo early labor will undergo an early wave of senescence. This is in the placenta. And likewise, women who undergo late labor have a late wave. And of course, we now know, certainly in mice and other organisms, that this secretory phenotype is very important for optimizing tissue repair and wound healing. So the way we really have to, I think we must think about the senescent phenotype is that it is an evolutionary balancing act. And this will be very important when we think about interventions, especially drugs, that will intervene in this very complicated phenotype. So there's good news, there's good things that senescent cells do, and then there are the bad things which we know are important for driving aging phenotypes. I'll summarize that data in a moment. So what we know so far about senescent cells in virtually all species that have been examined is based on biomarkers. There are now, this is just a partial list of the many biomarkers that have been used to identify senescent cells. Part of it includes what the cells secrete, but part of it includes things that change intracellularly, including the expression of tumor suppressor genes like P16 or P21. The problem is none of these markers are absolutely specific. And so for now, in order to really say that a senescent cell occurs in vivo, whether it's in a mouse or a human, we really need to rely on multiple markers. And doing that, many labs have now used combinations of these markers to ask when and where do senescent cells occur. And all of you probably know this, but they're seen in two physiological contexts. The first is they increase with age. And to my knowledge, this has been shown for virtually every vertebrate species that has been examined. And there will probably be some papers coming out in the next year or so showing that even in selected invertebrate species like Drosophila, for example, there may be certain tissues that seem to acquire cells with a senescent phenotype. Certainly in vertebrates, they increase with age in virtually every tissue that has been examined, including tissues we typically think of as being postmitotic, like the brain. So this panel here just shows human skin in young donors and older donors, and you can see that senescent cells are present in both the dermis and the epidermis. I do want to point out, though, that this last panel, which shows a string of senescent cells, is rare. We chose that actually as a kind of exception. For the most part, senescent cells are rare. They always are rare. I think the highest level has been maybe around 10% that has been validated by multiple markers. In addition to increasing with age, they are also present at sites of multiple age-related pathologies. So for example, in atherosclerotic plaques, there are often senescent endothelial cells. In osteoarthritic joints, so not rheumatoid arthritis, which is not age-related, but in osteoarthritic joints, you can find senescent chondrocytes. You find them, they're very prominent in the visceral fat that accumulates with age. And you find them in the brain, especially in the brains of patients and animals with neurodegenerative diseases. So here's an example. This is from Claudio Torres's lab. These are, these are senescent astrocytes present in the brains of patients. This is autopsy material, obviously, with Alzheimer's disease, and there are many fewer of them in age-matched controls. So the idea is that with age, we accumulate senescent cells. And we believe that due to this secretory phenotype, the cells secrete molecules that can cause neighboring cells to fail to function. And as a consequence, this could explain some of the degenerative diseases of aging. Of course, what happens with age is we're all accumulating premalignant cells. We all, even very young people, have some premalignant mutations. And we would argue that this secretory phenotype can even then drive these premalignant cells into full-blown malignancy. So this can explain a large swath of age-related diseases. Now, I also told you, you absolutely need this response in order to develop normally, in order to be born normally, in order to heal wounds. And so what do we do about this? How do we manage to suppress the bad things that this evolutionarily selected phenotype does while preserving the good? And so that gets then to the topic of this conference, which is this relatively new class of drugs, and also some natural products that can modify the senescent phenotype. And they're basically called two types of drugs. So xenomorphics are drugs that change the phenotype, selectively change the phenotype of senescent cells. I'll show you an example of that. And the other are the so-called senolytic drugs, which are achieving much more attention because they have been shown to selectively kill senescent cells. And as I pointed out, since these cells are always in a minority, there's less fear that there will be damage if you get rid of them because you're not depleting tissues of lots and lots of cells. So in thinking about how to modify the senescent phenotype in order to prevent its pro-aging aspects, the first thought that came to many of our minds is to stop the cells from secreting. We know that a lot of the aging phenotypes are caused by these pro-inflammatory cytokines and growth factors and DAMPs that the cells secrete. And so what if we can get them to stop secreting, or at least partially stop secreting? And this is the class of drugs that have been termed xenomorphics. They work, they do work. We have used some of them, and a number of other labs have developed other xenomorphics that they've tested on various phenotypes. There is a problem with this approach, though, and that is the targets. So when we first suggested to ourselves that maybe this would be a viable approach, what we did was we drilled down into the cell and molecular biology of what drives the secretory phenotype. And we found major pathways that drive these secretions. And the good news is that these pathways were already known. And the even better news was there were drugs that inhibited these pathways already developed by big pharma and even small pharma. So here are the three pathways that we've discovered. There are others as well that other labs have discovered. So one is the DNA damage response pathway. For example, if you inhibit ATM or CHIP2, you can prevent secretion of at least the pro-inflammatory arms of the secretory phenotype. The other is this P38 MAP kinase NF-kappa B pathway, absolutely essential for an early response to wounding. But if you inhibit this pathway, you can again suppress a lot of the secretions. And the third is this pathway you all know, the mTOR pathway, which is inhibited by rapamycin and rapalogs. And this mTOR pathway is, of course, essential for the response of cells to nutrients and growth factors. And that's exactly the problem. These are all important pathways that are required for normal tissue homeostasis. And so the last thing we want to do is to chronically inhibit these pathways. And it does have to be chronic because, as I said, there are drugs. We add the drugs, the cells stop secreting, that's great. But then we take away the drugs and the cells start secreting again. So that means you need constant pressure from the drug. And I would argue there is just no such thing as a perfectly safe drug that you would want to give for long periods of time to otherwise normal people. So although this is still a very promising approach for certain indications and for short-term solutions, we thought there needed to be a longer-term solution, and that was to try to selectively kill senescent cells. And this is the basis now for this relatively new class of drugs called senolytics, which is growing almost by the day. So the first approach to testing the idea that selective killing of senescent cells might be beneficial was done with a transgene. So obviously this is not a drug. This is something that can't be done immediately in humans, but it can be done in mice. And there were two mice that were developed, one by our colleagues at the Mayo Clinic, Jan van Dersen and Jim Kirkland, and the other by us. And they both took advantage of the fact that senescent cells often, not always, but often express P16, the tumor suppressor P16. And so what we did is we made a transgene So this is not a knockout or not partial knockout. The animals have normal copies of the endogenous P16 and P14 genes, which are contiguous. It's a very complicated locus. But we engineered a bacterial artificial chromosome that we could then have the promoter drive an artificial protein, which is a fusion protein between a luciferase, so you can image senescent cells in vivo, a red fluorescent protein, so you can sort the cells from tissues. But most important, this viral thymidine kinase, which has a very high affinity for the prodrug ganciclovir, turns it into a phosphorylated derivative that is a DNA chain terminator, which in senescent cells enters the mitochondria, fragments the mitochondrial genome, and causes the cells to die by apoptosis. So between our transgenic mouse and the mice that were developed by the Mayo group, these mice have been shared with, I can't keep track anymore, but literally 4 dozen academic labs who have then used these mice to ask, do senescent cells contribute to their favorite disease, depending upon what they were studying? So what this slide shows is a summary of published results. This is actually already out of date. I can hardly keep up with the literature right now, but you can see that virtually all of the diseases that I had on that first slide are in some way impacted in a positive way by eliminating senescent cells with these transgenes. In some cases, what eliminating senescent cells does is it delays the onset of the pathology. In other cases, it reduces the severity of the pathology. And in a few cases, for example, osteoarthritis, it actually can partially reverse the pathology. So given the fact that this worked so well in a transgenic mouse background, of course, this gave birth to this new class of drugs that are proposed to selectively kill senescent cells only. So selectively is in quotation marks because the selectivity is still not been established. In fact, there are drugs that act on some pathologies but not on others, and some natural products likewise act on some but not on others. But we need to understand this better. We need to understand the selectivity better. So let me summarize for you what the big questions are in the field with regard to senolytic drugs. First is, to my knowledge, there is no single senolytic drug that kills all known senescent cells. Among the drugs that are termed senolytic, that is, they have a selective ability to kill some senescent cells, they include both synthetic compounds as well as natural compounds. Very interestingly, many of the synthetic senolytic drugs are failed anti-cancer drugs. And this is interesting because there's a huge difference between the strategy behind killing senescent cells and killing cancer cells. If you want to cure cancer, you pretty much have to kill every cancer cell. Otherwise, the cancer will recur. But we now know from the transgenic mouse models, as well as from the mouse experiments, that it really is not necessary to kill every senescent cell. If you can reduce the burden of senescent cells by 70 or 80%, you can have remarkable positive effects on health span. So I want to give you an example of the heterogeneity of senescent cells, which is part of the problem why we haven't found a single target that is druggable. And that is that senescent cells are really surprisingly heterogeneous. So what I'm showing you here is single cell analysis comparing non-senescent normal human cells, these are fibroblasts, with their senescent counterpart. In this case, they were induced to senesce either by a genotoxic insult like ionizing radiation or a chemotherapeutic drug like doxorubicin. So this is P21. This is very commonly expressed by senescent cells. There are very few P21 positive cells in the non-senescent population. I should point out that when you talk about primary cells from humans or mice, they're never 100% non-senescent. There are always a few senescent cells that occur just by virtue of culturing these cells, but still very few P21 positive cells. If you now look at the senescent population, what you can see is that there are many more P21 positive cells, but there are groups, for example, this group here, that are far less than some of the other groups. And so again, targeting P21 will eliminate probably many of these cells, but there will be cells that will appear to be resistant. This is even more apparent when you look at the expression of this gene, MICA. MICA is a ligand for natural killer cells. It's not expressed very much in non-senescent cells, but there are populations of senescent cells that express high levels of MICA. There are actually 6 ligands for natural killer cells, and 4 of them increase in different populations of senescent cells. And we think this is one of the body's ways of eliminating senescent cells by allowing natural killer cells to attack them. But then there are subpopulations like this population here, which just simply don't express this gene. And they don't express many of the other genes that are associated with natural killer cell susceptibility. So again, we have to get a handle on the heterogeneity, and we have to understand whether some of these cells are good guys or bad guys. We just don't know. This is a big unknown in the field. Of course, the secretory phenotype, but actually many of the phenotypes of senescent cells varies with cell type. So there are no 2 cells that are identical. This is an early example of an antibody array showing fibroblasts and epithelial cells from the same individual. So this is the same genotype and the same tissue. This is from a human prostate. And you can see that there are some secreted products that are stromal specific and some that are epithelial specific. And then more recently, many labs, including ours, have done RNA-seq. And you can see, for example, if you compare human astrocytes and human fibroblasts induced by the same insult, ionizing radiation, there's overlap for sure, but there are distinct cell type specific differences. The other thing that the field has to deal with is that the senescent phenotype is not static, and this is especially true for the secreted products. So what I'm showing you here is a time course of a gene that codes for a pro-fibrotic leukotriene. So the cells, these are normal human cells again. The cells are induced to senesce by ionizing radiation. You get a huge increase in the expression of this gene. It then declines. It then rises again, but then slowly declines eventually to baseline. This is very different from the expression of this prostaglandin gene, which is anti-fibrotic, where you can see there is no early response, but there's only a late response that then continues on for many days and actually many weeks afterwards. I will remind you that you need this pro-fibrotic leukotriene in order to undergo wound healing. And then you need this prostaglandin in order to resolve the fibrosis of normal wound healing. And we recently showed that patients with idiopathic pulmonary fibrosis are very good at inducing the pro-fibrotic gene, but very poor at inducing this anti-fibrotic gene. So we think this will have clinical relevance when we start targeting these cells. And then finally, I do want to emphasize that the secretory phenotype, which we think causes most of the aging phenotypes, really varies depending upon how the cells became senescent to begin with. And another big unknown in the field is we simply do not know what drives cells into senescence in vivo naturally during natural aging. This is still a big unknown. A little bit of good news, though, is that we recently published a paper showing some of the core secreted products and compared it with data from Luigi Ferrucci's lab, in which he looked at biomarkers in the plasma of humans as they age. And there is overlap in these two secreted products. And this is just our top four hits, but there are about 40 proteins that are shared between the secreted products of senescent cells and those that are found as aging biomarkers in humans. And so I will just summarize by saying, from everything we've learned, it seems like what evolution selected for is the transient presence of senescent cells, which are absolutely required for optimal tissue repair and tissue homeostasis. With age, though, some of these senescent cells persist, and they can then go on to undo the good things and drive age-related diseases. And the challenge for us is to develop selective identifiers of these two types so that the drugs we develop can, of course, be developed to be more specific. So I'm very optimistic that these senolytic drugs that are on the horizon really hold promise for extending a health span of humans. But there's a lot more work we need to do to understand how they differ and how they can be selectively targeted. And I will stop here. And of course, everything I say is due to a village, and the village includes present lab members, past lab members, and an enormous group of collaborators who have helped us move this field forward. So thank you.
Judy, thank you very much. Big round of applause for your wonderful work, for the wonderful talk, and for the many entrepreneurs who you inspired to go into senolytics and xenoremediators, modulators, antifibrotics. So it's just you spurred the industry. And I remember, just 16 years ago, you would come to a conference and people would still take the SASP serious. And now it's, you probably know that what we do, right? So we work with many big pharma companies, and many big pharma companies also have senolytic programs, just as a so yeah, so everybody is looking at that, and great work that you are doing. I apologize for bringing this question, but it's currently the most upvoted one. So, and I'm gonna read it verbatim. So thanks for a great talk, Judith. I think that we're all wondering about this tough question. Why do you think the recent Unity trial failed?
Yes. So I don't know how many of you know Edward Lamphier, but he was the CEO of Sangamo Biosciences. I was actually on his board for a little while. And he's famous for the following quote: "There's no such thing as a bad drug. There are only bad trials." So, I mean, of course there are bad drugs, but Edward was making the point that everything depends on how the trial was designed. So I will remind you how that osteoarthritis trial was designed: a single dose and a set time point. And so they're going back now, looking at other doses and other time points. I'm not pessimistic about the promise of that drug, but I do think that the problem was probably the trial as opposed to the drug. I hope.
So considering that you have multiple targets in the pipeline at Unity, and this one, the first one, read out negatively, and again, many people ask, why did it get progressed into phase 2 when the results from phase 1 were, let's say, not as optimistic? What do you think about, if you were to compare the program 1 and program 2, so the BCL-XL targets that you have in currently, I think, preclinical. If you were to compare those two targets in terms of their potential senolytics, potential targets for senolytics, which one would you rate better?
Yeah, that's a good point. So we have worked with some of the BCL family member drugs that target those family members in mice and human cells, but not in humans, of course. And what I can tell you is they are also extremely variable. Some tissues respond, some tissues do not. It could be a delivery problem. We haven't done good pharmacokinetics. So I think it's still very early days at this point. Also, we don't know whether cells that overexpress those anti-apoptotic proteins are the bad guys. We still haven't proven that. So I think there's a lot more basic, of course, I'm speaking now very self-servingly. I'm a basic scientist, but I think there is a lot more basic science that needs to be done.
Agreed 100%. But again, rooting for the program number 2. And I know those molecules and I know where they came from. Very promising. Yeah. So a question from Sarah Sepe, very highly ranked question. Regarding cell specificity senescence, do you think that neurons can undergo senescence? Have you ever observed neurons positive for senescence markers?
Yes, that's a great question. If you asked me that question 2 years ago, I would have said, oh no, they're already postmitotic. You know, I've totally changed my mind. And I want to give credit to Carl Harrop, who took me aside at some meeting and showed me data of neurons that were expressing a SASP that looked a lot like we were observing in astrocytes and fibroblasts. Things like upregulation of certain tumor suppressors. So I think we need to back off on that stance and say, yes, I think it is possible. The question is, will those neurons be targeted by senolytics? And that, I think, is still an open question.
Thank you. And big question from Phil Coriel. Senolytics or xenomorphics? Which do you think have the best potential for treating senescence-associated diseases?
Oh, I think senolytics, as I pointed out with xenomorphics, unless they're natural products, but even then there's a lot of, any drug will kill you if you take it long enough and at high enough dose. I'll remind you, you'll die if you drink too much water. So I think drugs that can act in a hit and run fashion make more sense to me.
Got it. Same opinion. So Cyril Lager asks a question. Thanks for the talk. In your single cell data, does the expression of P16 also correlate with senescent or non-senescent cells, as you showed on for P21?
Yes, yes, meaning that there are cells that express high levels of P16, cells that express lower levels of P16, and then within the senescent population, there are P16 negative cells. Now, let me just say one thing about single cell analyses in general. It's a snapshot in time. And if a gene is oscillating in time, that snapshot could really be capturing cells that are expressing, but not the cells that are not expressing. It's a big caveat. I know there are computational ways now that are being developed to overcome this problem, but it is a problem. So we can't take the single cell data as being static in time.
Thank you. Great answer. A question from Emery Targiolu. I hope I'm not mispronouncing this in a very bad way. So great information. Has there been any studies on biomarkers that can categorize different types of senescent cells, like trying the markers in the Ferrucci study with senescent cells that have specific common transcriptome? So are there any specific drugs that would target those specific types of senescent cells using transcriptomic biomarkers?
So to my knowledge, the answer is no to both questions. We don't yet know how to take plasma and say, this person has high senescent endothelial cells as opposed to, we just don't know how to do that. And as far as selectivity of drugs, I don't know that we're there yet. Maybe there's unpublished data, but certainly we don't have any evidence for that yet.
Thank you. And what would be your piece of advice to everybody who is in the field of senolytics and considering going clinical? I mean, there is an entire plethora of those entrepreneurs who are considering taking some of those senolytics into clinic. What would be your advice?
Learn as much as you possibly can about your drug. It's good as well as it's bad. Don't shy away from the bad. And of course, first and foremost, do no harm.
Got it. And one kind of question from my side. It looks like there are multiple compounds that have possible geroprotectors that might have, the senolytic effect, but also they are targeting other hallmarks of aging.
Yes.
It looks like some of the, even rapamycin could be possibly senolytic. Can you comment on that? And can you comment on any of the compounds that have, more than one hallmarks of aging kind of direction covered in addition to senolysis?
Yes. So let me be the first to say, I don't think we can explain all of aging. We certainly cannot explain species-specific lifespan by senescence. I mean, there are other processes that are driving aging and limiting lifespan. They have yet to be discovered, I think. As far as drugs like rapamycin, we don't think it's senolytic, but we do think it's xenomorphic. And we've shown that. And what the beauty of rapamycin in my mind was that it inhibits a positive feedback loop. And when that loop is inhibited, when you take away the drug, it takes time for the loop to be reestablished. So it's probably an example of a xenomorphic that doesn't need to be constantly present. And I'm more enthusiastic about those xenomorphics than the ones where you need constant pressure from a drug.
Thank you, Judy. Great talk. You're an inspiration to us.